Hi, thank you for joining us for the 10th Fkmcd Oxytec Public Educational Webinar. Our topic is preparing for the Fkmcd Oxytec pilot project, overview of field design and management. Our panelists are Andrea Leal, Executive Director of the Fkmcd. I'm Meredith Vinsum, Oxytec's Head of Public Affairs and a Florida native. I also relocated to the Keys last week. Happy to be here. And we have Nathan Rose. Nathan is Oxytec's Head of Regulatory Affairs. We're hosting this series of public educational webinars to share information with residents of the Florida Keys and provide forums to answer questions. The webinars are open to everyone. Webinars are recorded and made available for everyone after the event on the Oxytec website, also on our project website, keysmosquitoproject.com, and we also post the recordings to our YouTube channel. All questions relating to the webinar topics will be answered. We may group similar questions together. If time runs out, we will accept questions in writing via florida@oxytec.com. Questions and answers will be published in writing after the event with links to external or related online resources. We'd also like to draw your attention to our upcoming webinars. In February, we'll host a roundtable discussion about controlling the Aedes aegypti, the vector of dengue, Zika, heartworm, and other diseases. And in March, we'll look at community partnerships and the role communities play in our pilot project. Our agenda is preparing for the Fkmcd Oxytec pilot project, an overview of field design and management, and then we'll spend the second half of the webinar, so 30 minutes, taking your questions and answering them. You can begin to enter questions in the chat box and we'll address them then. Now I'll turn it over to Andrea.
Thank you, Meredith. I think I always kind of like to start out by answering the question that we get most often, which is, why are we looking at this technology? Why the Florida Keys? And specifically, why right now? One of the biggest reasons that we're looking into these alternative technologies is because we've seen a rise in mosquito-borne diseases in South Florida within the last decade. So back in 2009 and 2010, we saw dengue fever in Key West, just under 100 cases transmitted by our mosquitoes. And it was the first time we've seen that since the 1930s. And then fast forward to this last year, and we saw 67 cases of dengue fever in Key Largo that were locally acquired as well. And then not to mention in 2016 with the Zika outbreak that occurred in Miami-Dade. So we've really been looking at how to control these specific mosquitoes that are causing these diseases for the last 10 plus years. And it's always right there on the forefront of our minds for mosquito control. And one of the biggest issues that we've seen with the mosquito that does transmit these is we are seeing insecticide resistance. So, the products that we currently have at our fingertips are not working like they used to. And we're just seeing that resistance building over time. And so unfortunately, conventional tools are not working as well as what we really would need them to be. So it's really turning us on to, looking into this Oxytec project, looking into alternative ways to control this mosquito since it is so difficult to control. Another major consideration that the district always tries to keep in mind is, the Florida Keys are located entirely within a national marine sanctuary. So we're very environmentally conscious. We want to make sure that what we're doing not only protects public health, but also the environment. And this specific tool works on mosquitoes only. So, we don't have to worry about potential non-target effects. We're really only looking at affecting this one mosquito species. And we have really great working relationships with our local land managers, both state and federal. So we want to make sure that we're doing the best that we can for all of the species here in the Florida Keys. Next slide, please. So this specific technology works on the Aedes aegypti mosquito. And like I mentioned previously, the Aedes aegypti mosquito is the one that's responsible for transmitting dengue, Zika, chikungunya, yellow fever, and is not from here. Basically, it is originated in Africa, came over a couple hundred years ago, and we've seen it really spread across the globe, causing these diseases also to spread across the globe. So we're really looking at controlling an invasive species. And again, this technology is specific to that one species and not the naturally occurring species that we see here in the Florida Keys. I think with that, I'll go ahead and turn it over to Nathan to get into the project a little bit more.
Okay, thanks very much, Andrea. So what I'd like to do is just start briefly by talking about what the Oxytec mosquito technology is. And then I'll talk a little bit more about how this technology works to actually suppress a mosquito population. And then from there, I'll move on to talking about what the field projects will look like. So Oxytec makes male-only Aedes aegypti mosquitoes for release. And male mosquitoes do not bite. But what they do is they fly out and they mate with wild females, and they pass on to those wild females two genes. One of those genes is a self-limiting gene, which basically stops all of the female offspring of those matings from actually developing. And the other gene is a fluorescent marker gene, which allows us to actually track and trace our mosquitoes in the field. And because this is a mating-based technology, it gives completely targeted suppression only of the Aedes aegypti species, because this cannot mate with any other species. And it is safe, it's non-toxic, it's non-allergenic, and it has proven effectiveness in the field in several years of releasing these mosquitoes in other places, particularly in Brazil. And I'll talk about that a bit more in one of the coming slides. So on the bottom right-hand side of the slide, you can see this fluorescent marker gene, which allows us to identify these mosquitoes very easily. So you can see that whether it's a larva or a pupa or an adult mosquito, we can see the fluorescent marker gene and track and trace these mosquitoes in the field. And then on the left-hand side, you can see a little bit about how these mosquitoes are deployed, and I'll talk about this more in the coming slides. But the basic idea is that we have a very low-tech egg-based deployment of these mosquitoes. We have a box, we take these mosquito eggs, we put them in the box, and we add water. And after a few days, we start to see these male mosquitoes hatching and emerging from this box. But no female mosquitoes are produced and actually fly out of this box at all. So how does this self-limiting gene work? What it does is it stops any females that carry this gene from surviving to adulthood. And the male mosquitoes which have this gene are unaffected, and they can pass on then another copy of the self-limiting gene to their offspring. And so we can achieve significant suppression over multiple generations of these mosquitoes. The other thing that this self-limiting gene allows us to do is to produce only male mosquitoes using these boxes in the field, because the females can't actually survive if they carry this gene. And that means that we have a massive reduction in the complexity of actually producing mosquitoes for release, because we actually make those mosquitoes in situ in the field. And we have released at least 20 million of these male OX5034 mosquitoes already in Brazil. Globally, Oxytec has released around 1 billion mosquitoes, including its older technology, OX513A, in multiple different countries, Brazil, Cayman Islands, Panama, and other parts of the world as well. And in all of these releases of over 1 billion mosquitoes, we've never seen any negative impact on either human health or the environment over a period of about 10 years. So this slide here shows some of the demonstrated effectiveness of this mosquito in Brazil. So on the right-hand side is a graph just showing a pilot project that we carried out in a city called Indaiatuba, which is near Sao Paulo in Brazil. And this was a 17-week treatment with mosquitoes, so releasing these mosquitoes for about 17 weeks. And what we saw was that within about 6 weeks after we started releasing these mosquitoes, we saw a significant reduction in the abundance of the wild mosquito. And within about 12 to 14 weeks, we'd achieved more than 90% suppression of the wild mosquito population by releasing these male-only mosquitoes. So they were going out, they were mating with wild females, none of the female offspring was surviving. And as a result, over just about 3 months, we saw the population of the wild mosquitoes crash. And that was a lot faster than we expected. We were expecting it to take about 3 months before we started to see any effect, but this actually worked really, really quickly. Within about 6 weeks, we had significant suppression. And throughout this project, we didn't see any unintended impacts. We had only males being released as planned. And additionally, this gene is fully self-limiting, and that means that after we stop releases, there's no long-term persistence of these genes in the wild. Massive reduction in the operational complexity because we were releasing these mosquitoes from boxes, and also very strong public support in Brazil, where dengue is an ongoing problem year upon year. It returns to many cities in Brazil, and it causes significant health impacts. And so in that context, we had very strong public acceptance of this project. So moving on to what we plan to do in Florida. I'll briefly talk about how we actually get these mosquitoes from where we produce eggs in the UK to Florida. And then I'll talk a little bit more about how the box actually works as well. So in our production facility in the United Kingdom, we produce these OX5034 mosquito eggs, and we package them into little capsules. We then ship those across to Florida. And once they're in Florida, we take them and we put them into these mosquito release boxes, which you can see on the right-hand side there. And in those mosquito release boxes, we have the eggs, we have some diet to help the mosquitoes as they grow, and then we add water. And as I said, within a few days, around 10 days, we start to see these male mosquitoes emerging from these boxes, going out into the wild and finding the wild females to mate with them. We don't release any females, and we verified this with multiple seasons in Brazil and also with extensive lab validation of this as well. And because it's only the females that can bite, we're not actually releasing any biting mosquitoes either. There are only male adults that are produced in this box. And additionally, there is no use of tetracycline in the box to actually produce these male mosquitoes, and there's no tetracycline usage in Florida whatsoever. And these boxes are typically placed in out-of-the-way areas. That might be on a pole, that might be on a wall, but we want to make sure that they're not intrusive in the environment, so that those male mosquitoes can actually not breed, but they can develop, and then they can be released. Okay, so briefly, this is one of the questions which we're often asked by communities where we release these mosquitoes. Do the mosquitoes that we're releasing from these boxes actually bite anyone? And the answer is no. Only female mosquitoes can bite. Only females have the mouth parts to actually allow them to bite humans or other mammals and to take blood. Males don't have these mouth parts. They actually they feed on nectar. They're not able to feed on blood whatsoever. And so because we're not releasing any female mosquitoes, there is no possibility of the Oxytec mosquitoes actually biting anyone. And these Oxytec mosquitoes are completely safe and non-toxic. And that's a conclusion that's been reached by 9 different agencies in the US that reviewed this project, and also other regulatory agencies worldwide. So I'm going to move on now just to talk about how Oxytec actually manages its field projects and the kind of data that we collect and what it might look like in the field. So there are 3 main types of projects that Oxytec has done historically. The first category is what we call regulatory trials. And these are small projects with high statistical power. These are protocols which are reviewed and approved by regulators. And they're assessing both the biology of the mosquitoes released, but also their efficacy in actually mating with the wild mosquitoes, causing their offspring to die, and so on. And this is really what the project in Florida will be focusing on. These are small-scale projects focused on generating data for regulators to be able to assess performance in the field. Once we've done those, we'll often move on to larger-scale demonstration trials, and these are larger pilots to demonstrate area-wide performance, so maybe over a whole suburb in a city. And those are often also designed with regulators, and they will have a comparison with the control area where we don't release any mosquitoes. And then finally, Once we have full regulatory approval and freedom to operate, we'd move to larger scale operational deployment, where this is actually used by mosquito control authorities as a vector control tool to help suppress these mosquitoes over a larger area. An important thing to say about this mosquito is that it basically performs like a larvicide. So larvicides are already used by mosquito control to be able to kill mosquito larvae where they're breeding in water. And that can be aerial larviciding, it can be mosquito dunks, which are placed into water to kill larvae that are developing there. And this effectively works in a similar way because we are killing female larvae of the next generation, making sure that they're not able to develop and survive and become adult females. So when we're actually doing a project in the field, there's several different metrics that we tend to assess. We want to look at the overall abundance of mosquitoes. And the way we would do that is we would look at the number of wild Aedes aegypti mosquitoes that we catch in traps. And that allows us to actually check what the baseline population is before we start releasing mosquitoes, and then to measure any changes that happen as we release our mosquitoes. We'd also look at what we call an overflooding ratio, and that is the ratio of how many male mosquitoes we've released to the number of wild male mosquitoes. So for example, we might release 5 times as many males as there are wild males. And that helps us just to measure and achieve the optimal dose rate to really be able to impact the local mosquito population. We'll also look at something called mating fraction, and this is the proportion of the wild females that have actually mated with one of the males that we've released. And that helps us to evaluate the proportion of the wild population which has been treated by our mosquitoes. And then finally, we'll look at efficacy, and this is the percentage of treated females that die. And here we're really just confirming that the gene which is being put into these females through mating is 100% effective against the female offspring. So the way we collect data in the field, we use 2 types of traps, and the first of these is what we call an egg collection trap or an ovi trap. And these are small plastic cups that have a little egg-laying surface in them and some water. And this allows us to monitor the number of eggs that are laid by the wild females. So those wild females might have mated with one of our males, or they might have mated with a wild male. And what we can do is we can take those eggs back into the lab and we can hatch them, and then we can monitor the larvae as they develop and see whether or not they come from mating with a wild male or one of the males that we've released. And so we have lab-based monitoring using microscopes, examining each of these larvae that develops, and that helps us to track performance, but it also helps us to track the quality of the mosquitoes that we're releasing. The other thing we'll typically do is we will have traps that trap adult mosquitoes. And there's a few examples of these on the left-hand side there. They come in all different shapes and sizes. But typically what these do is they will lure wild mosquitoes, males or females or both, into a trap using some kind of bait or some kind of lure that might be carbon dioxide or some other pheromone. And then we can go and collect those adult mosquitoes. And that helps us to monitor the number of Aedes aegypti adults in the area, and also to monitor those ratios of our mosquitoes to the existing adult mosquitoes in the area. And so as this project kicks off, we will start to have both ovi traps and adult mosquito traps in the locations where we start the mosquito releases. And later on, we'll talk about how you can actually get involved in this and maybe request to have one of these traps in your property to help with this project. So as Andrea said earlier on, this project is really about helping to broaden the toolbox that mosquito control authorities are using to protect communities against this invasive species and the diseases that it carries. And it's really important that in this project, we're preserving the quality of life for residents and the delicate ecosystem in the Florida Keys, and also helping to evaluate a technology which can do both of those things. And so there are several different parts of this project. There is already a lot of community engagement happening right now, both on the ground in communities, but also through these webinars and through various other avenues that we use just to try and inform everyone as to what this project will involve. And then there are 3 different types of releases that will be happening in this project. So the first one is what we call a mark release recapture experiment. And this is basically where we release male mosquitoes and we will then recapture those to measure how far they've flown and how long they've actually survived in the local environment. And then we have 2 parts of the project, Project A and Project B, which I'll talk about in one of the coming slides as well. Overall, throughout this project in the Florida Keys, we're expecting to place about 130 of these boxes over a period of about 28 weeks. And throughout, there will be no females released, only male mosquitoes that are actually being released. So, two main components to this project. And on the left-hand side is Project A, which is really a single point release. And what we'll be doing here is choosing a few locations, putting a single box in the environment in 9 small areas, and basically releasing mosquitoes over a period of about 12 weeks. And this will allow us to see how far these mosquitoes are going, how long they're living, how effectively they're mating with the wild females, just in the vicinity of that single box. And then the second part of this project, Project B, is a multiple point release. And so here we would have a small number of devices sort of scattered over a small neighborhood. And we would do this in up to 6 areas for about 4 months in total. And this will allow us to evaluate how effective these boxes are at releasing mosquitoes steadily over a whole neighborhood and being able to actually mate effectively with the wild mosquitoes in a larger area. And so these 2 parts of the project are really what we're planning to focus on in Florida. And you can see on the right-hand side there, one of our project scientists placing one of these boxes in the field, putting some water in, adding the eggs, and really getting that ready for deployment. And the project, as I've mentioned, will have these single multiple point releases. We'll also have replication, making sure that we're doing the same thing in multiple locations so we don't have an area-specific effect. And we'll have comparison to untreated areas. So locations in the Keys where we're not releasing any mosquitoes, but where we're still monitoring the wild mosquito population so that we can compare that with the locations where we release our male mosquitoes. The specific locations are still being determined, and the timing is anticipated to start in the 2021 mosquito season. And that'll be preceded by some baseline monitoring. So basically monitoring the wild mosquito population in all of these locations for several weeks before we start the releases. In addition to looking at things like the male flight range, the longevity of the mosquitoes, how long the effect lasts, we'll also be looking at natural breeding sites to see if our mosquitoes are able to mate with wild females that are then laying their eggs in natural sites, so pools of water that might be present in people's backyards. And then also the things like the percentage kill of the female mosquitoes and the percentage of the wild population which are actually treated by these male mosquitoes which are being released. And so this slide here gives an overview of the whole project, sort of pre-season, the main mosquito season, and then post-season activities. So from the very outset in the pre-season, there is community engagement going on, and this will continue right the way through the mosquito season and afterwards. And this is really an opportunity to make sure the community is fully involved in this project, can ask any questions, can give any input, and can also help to host the boxes that release mosquitoes and the traps that help us to monitor them. In addition, there is staff training and preparations for the project, which are well underway right now. And I'm sure that people in the local community will be meeting some of the Oxitec and the Fkmcd staff that will be involved in this project. And then, as I've just mentioned, there's also pre-release mosquito monitoring. And this is just to help set a baseline of how many mosquitoes are in each of these locations, what's their behavior like, where are they found. And that also helps to inform the sites where the releases will actually happen. Then once the mosquito season kicks off, we'll have the release of Oxytec mosquitoes. And throughout that time, there is ongoing monitoring of these traps to look at what happens to the mosquito population, evaluation of the data, and making sure that these releases are happening effectively. And then in the post-season period, after we stop releases, we'll continue to monitor the environment for many months afterwards to basically look for the disappearance of the released mosquitoes and the genes that they carry from the environment. And that's a really important part as well, to evaluate the ongoing impacts there. So I'm going to hand back to Meredith at this point, who's on mute.
Thank you, Nathan. We've got just a couple of slides left to review our recent community and engagement efforts and also opportunities to join the project. You know, COVID has caused us to be creative with how we engage the community, and We've done now 10 of these webinars. We also do virtual presentations for groups in the area. Some of you may have begun receiving postcards in your mailbox. We've released a virtual tour of our Oxytec labs in the United Kingdom, in Brazil, and here in Marathon, Florida. We've done some in-person events, and we are going door to door. I was going door to door last week, and we had teams out yesterday and this afternoon. So we are doing that in a safe way, knocking once, wearing our masks, stepping back 6 feet. And a number of people, they are answering the door. We weren't sure if they would, and there is interest and there are questions about the project. So we like to say that we're transitioning more now from the screen back to the front porch, and we'll also be going to more of the farmers and artisan markets in the community, just to be able to see people and engage with them in a safe way outside. Nathan mentioned that there are options for local residents to join the project. We've set this up on our project website, keysmosquitoproject.com. And you can join the project, and you can sign up either to request a mosquito box for your yard or a trap. As you heard, those are really important for monitoring and for the success of the project. If you request a box or a trap, we can't guarantee that you'll get one, but if we come to your neighborhood, it will be helpful to know who there would like to be involved. You can also sign up for updates. We have a listserv and usually send out about one update a week, or you can volunteer as a project ambassador. And now we will begin to take questions. Okay, the first question is, how can you be sure, and Nathan, I think I'll send this question to you. The question is, how can you be sure that only male mosquitoes will be released?
Okay, thanks for that question. So I think two parts to this. One is that we have extensive data both from the lab and from the field in Brazil, where we've released these mosquitoes for more than 2 years. And in all of those, we have seen that this gene is completely effective, 100% effective in ensuring that only male mosquitoes can survive if they carry this gene. And these data were shared with regulators. So that was the EPA at the federal level and also the Florida state regulators who approved this project. And again, that confirmed that only male mosquitoes are able to survive. And in addition, we have ongoing quality control on this mosquito strain as part of this project, ensuring that the strain continues to perform as it has done in the past. And so we are confident that this will be only releasing male mosquitoes in this project.
Thank you, Nathan. The next question is, and I'll take this one, is there interest in Oxytec from any other places in the US? And the answer is yes. We've continued to receive interest from a range of mostly local governments in the United States, and those are coming in pretty consistently. So the next question, Nathan, I'll give this one to you. It's how much time is in a generation from birth to reproduction to death?
Okay, that's a great question. So the Aedes aegypti mosquito, when an egg hatches, it'll typically take a couple of days, so it'll take about a week, for that egg to actually develop through the larval stages. And so that's when the mosquito is swimming around, it's feeding. After it's finished developing as a larva, it'll then turn into a pupa, and that'll typically live as a pupa for a day or two. And then once that pupa ecloses into an adult mosquito, then the life of the mosquito will actually depend on the environment that it's in. And so an average male mosquito might last just a couple of days in the environment. A female mosquito, a wild mosquito, can typically live for a bit longer, maybe a week, maybe a couple of weeks on average. And so a generation time on average is about sort of 3 weeks to a month. That will depend a bit on temperature. So warmer temperatures, you might find that the aquatic stages of the mosquito, the larvae and the pupae, will develop faster, but you might also find that the adult mosquitoes don't live as long if it's really, really hot. And so on average, we're looking at 3 weeks to a month per mosquito generation for the 82-tip mosquito.
Thank you, Nathan. The next question, Andrea, I'll send to you, and then Nathan, you may want to add on, but the question is, can you extend the project for extra seasons?
So right now we are under an experimental use permit from the EPA, which will allow us to basically go up until the spring of next year. What happens after these projects are completed? The data will be turned into the EPA, and then I'm sure there'll be extensive discussions on really what the next step is going to be at that time. So for now, we're hopeful that we'll get through both of these projects in this upcoming season and get some great data to really see what the next steps are.
Yeah, no, absolutely agree. If for whatever reason we wanted to extend the permits, that's also something which EPA can do. But as Andrea said, we're definitely hoping to get through all of this season.
Thank you both. Nathan, the next question is for you. And the question is, what happens when a male GM mosquito mates with the local female and the female takes her blood meal? Have you tested these mosquitoes for the contents of the impregnated mosquito's saliva?
Okay. So a female that mates with one of our males, the only thing that's going to be affected in that female is the eggs which she carries, which will be fertilized by one of the male mosquitoes that we release. And so those eggs which then contain the DNA of the female mosquito and of the male mosquito which we release. There's no mechanism in that mosquito for any of the male mosquito's DNA to make it into any other part of that female. So I'm not quite sure where this question is coming from, but the only impact on that female mosquito is actually going to be on the eggs which she is laying. And that's something which we assess. So we'll be looking at those eggs that we'll catch in the ovitraps, we'll bring them back into the lab and assess whether those eggs come from a mating with one of the wild males or with one of the Oxytec males. The wild females that are out there, they are definitely out there looking for people to bite and take blood meals, and that's how they transmit disease. But that whole process is not affected by their mating. The only thing that's impacted by the mating is the offspring. And as I've said, the female offspring that inherit these genes, they will actually die as very early stage larvae, so they will never become adult females.
Okay, thank you, Nathan. The next question, I'll give you a break, Nathan, and I'll answer this one. You've hosted 10 webinars. Do other pest control related companies provide this level of transparency? Not that we know of, but this is our approach. Okay, Nathan, this next question, it's for you. I hope it makes sense. Do the mated female mosquitoes also get controlled by the Oxytec males?
Okay, that's a good question. The answer is no. So a mated wild female that's mated with one of our males, she will go and lay her eggs, and then she will carry on and do whatever that female would normally be doing. But that female that gets mated is not going to die as a result of that interaction with one of our male mosquitoes. However, her offspring will. And so any female offspring that come from her will die. And that's really where we see the impact on the wild mosquito population, because if we are effectively killing all of the female offspring, we start to see massive reductions in that wild mosquito population.
Okay, thank you, Nathan. The next question I am going to send to you, and the question is, this is for you, Nathan. Mm-hmm. Have you read that mosquitoes in Africa were capable of taking 4 blood meals? Therefore, they were 3 times more capable of spreading malaria. These are the kinds of unintended consequences. It's the end of that sentence. Were these the Gates Oxytec mosquitoes?
Okay, interesting question. Oxytec doesn't have any mosquitoes in Africa. So Oxytec is working together with the Bill and Melinda Gates Foundation to develop a similar technology in mosquitoes that transmit malaria. But that is still in the lab development phase. We have not released anything in Africa at all. But the more important point is that we will not be releasing females either in this project with Aedes aegypti or in any of our work on malaria mosquitoes. The same principle applies, that we would only release male mosquitoes. And so the question about taking blood meals and what that might do to malaria transmission doesn't apply, because males are not biting, they're not taking blood meals, and they're not transmitting diseases. But taking multiple blood meals is not unusual. And so the really important thing is that we get rid of those female mosquitoes that are able to bite and transmit disease. And that's actually what this technology is designed to do.
Great, thank you, Nathan. The next question, Nathan, it's for you too. Is there any evidence that this technology can reduce disease?
Okay, great question. So definitely the aim of this technology is to get rid of the mosquitoes that are able to transmit diseases. And while we at Oxytec don't monitor disease directly, some of the cities where we've worked in Brazil have documented reductions, particularly in dengue case numbers in the areas where we've released our mosquitoes, when they compare them to other parts of the city where none of the mosquitoes were released. And so there is evidence from those municipalities, but it's not something that we'll be doing directly in this project in Florida.
Thank you, Nathan. The next question is relevant for both Fkmcd and Oxytec. So Andrea, if you could answer, and then Nathan, if you could too. The question is, how many staff are required for the project?
So on the district side, we will have staff working part of their time on this project, and we have, a good handful of staff that will work on each individual day. And I think it's very important that we're as heavily involved in the project as possible, in every single aspect, just so we're all comfortable with oversight, making sure we're following protocols correctly, et cetera. So like I said, on the district side, it's about a handful of staff that'll be working just part of their time.
And on the Oxytec side, so we have, I think right now, five or six staff in Florida already, including Meredith, our country director there as well, and then technical staff. But that's also supported by a lot more Oxytec staff in the UK and in Brazil as well, focusing on the production, quality control, quality assurance, regulatory, and so on. So on the Oxytec side, there's much more substantial staff numbers that are actually involved in making this project happen.
Thanks, Andrea and Nathan. Nathan, this next question I will send to you. And the question is, I know that Aedes aegypti is a non-native species in Florida, and you mentioned earlier that there were 0 reported side effects, but are there any negative side effects of the Oxytec mosquito deployment in locations where Aedes aegypti is native?
Okay, that's a really good question. So as we said on one of the earlier slides, Aedes aegypti originated in Africa. It's thought that it made its way across the Atlantic probably in the 16th century with some of the early Portuguese traders. It then moved from South America up to North America and then back across to Europe and into Asia, probably sometime in the 19th century. And all of those data are sort of backed up by genetic analyses of Aedes aegypti throughout the world. It's also been eliminated from many countries, and then it's reinvaded many of those countries again, unfortunately. But they are native in Africa. We would expect that these mosquitoes that we release would also be effective in controlling the wild mosquito populations in Africa. But that's not something that we've ever done up till this point. But it's definitely something which African mosquito control is very interested in, because dengue is a major problem in many African countries alongside a very heavy malaria burden. So again, there I think there is a real need for tools that can control this mosquito, whether that's an insecticide or whether that's a really targeted species-specific effect like the Oxytec mosquitoes.
Thank you, Nathan.
Meredith, if I can just add on to that a little bit as well, we're talking about a species that has really evolved into living in its own little niche. So bird bass, discarded tires, garbage that's laying around collecting water, and you're not finding, other species that could feed off of it living in this niche with it. Additionally, for us in the Florida Keys, we're looking at anywhere from about 2 to 4% of our entire mosquito population. So very minor portion of our mosquito population are these Aedes aegypti. So it's not really part of a natural ecosystem in many places. Now, I can't speak for the population in Africa, but I'm sure you find something similar in Brazil, Caribbean islands, throughout most of where you're going to find this species. So it doesn't have a lot of natural predators that are going to live in a discarded tire with it. And I think that is another thing, that we really focused on is eliminating it because of that evolution to really being next to humans and causing all of these diseases.
Thank you, Andrea and Nathan. I'll take the next question, and it comes in several parts. The question is, the boxes for releasing the mosquitoes, will they be reused? Are they biodegradable? And how many boxes are anticipated to be deployed in the field? The answer to the first question is yes, they will be reused. The answer to the second question, are they biodegradable? I don't think so. These are reusable boxes. We have interior components that are recyclable, and that part is replaced after the mosquitoes have grown there with their food and their water. And then the last part of the question, how many boxes are anticipated to be deployed in the field? For this pilot project, which has two components, Project A and B, we plan to deploy about 130 boxes.
And just to add to that, those 130 boxes will be put in place, but the inner part of those boxes, the liners, will be replaced once those mosquitoes have actually developed. We'll put in a new liner, new diet, new water, and so we can have a continual release from that durable outer box.
That's right. Thank you, Nathan. And the next question, Andrea, I think we'll send this question to you. The question is, will Florida develop a similar project to tackle other mosquito species? And there is a question here about the Culex. I'm not going to try to pronounce that. It's a very long word.
Culex mosquitoes they're a very common vector in Florida. Mostly West Nile virus are the concern when it comes to Culex mosquitoes, both Nigra and Quinca fasciatus, which probably one of those two are the ones you're trying to pronounce. But at this point in time, I don't know of any development into a similar technology for those specific species. However, I would imagine as we move forward with these projects and get great results, hopefully we see similar results to what we've seen in Brazil, that this technology will just continue to grow and be able to be used hopefully for other vector species as well.
Yeah, from a genetics perspective, I would expect that the genes would work just as well in one of the Culex species as they do in Aedes.
Thank you, Andrea and Nathan. Nathan, I'm going to send the next question to you. And the question is, how do you know how often a released Oxytec male is mating with females in the field?
Okay, that's a great question. It's quite a difficult thing to study to know exactly how many times they mate. But we would expect that our males would mate multiple times in their lifetime, but they will have a relatively short lifetime. So after we've released them, we'd expect they're only going to live a couple of days out in the field. And during that time, they'll have to find females and mate with them. What we do monitor is the offspring of those matings. And so we will look at the eggs that have been laid by the females that they've mated with, and we'll take those back to the lab and hatch them. And using that fluorescent marker that I showed earlier in one of the slides, we can tell whether or not their offspring have come from one of our male mosquitoes. And so that's really the easiest way for us to tell how effective our males are being in the field.
Okay, thank you, Nathan. The next question I'll take, and it says, very interesting indeed. This kind of tropical diseases are prevalent in developing countries with limited budgets, where cost effectiveness and budget impact models could be crucial for decision making. So that's not really a question, but I will say that, a difference between Oxytec's first generation, and that's the generation of the technology that we were working with when we were first invited to the Keys 10 years ago. It was similar in many ways to what we are releasing now, and we're proud of that technology. We stand by it. But that required us to rear mosquitoes to adulthood in factories and to then release them from bands. And this more recent method of deployment, to be able to use mosquito eggs in boxes and just add water and have the mosquitoes release themselves, it offers some operational benefits that translate into major cost savings and really accessibility. And that's the point of this technology. We feel a real duty to get it out to the communities that need it, and we want it to be accessible.
Could I just add something to that, Meredith? The really exciting thing about this version of the technology is that we can make the mosquito eggs in a central location, but we can ship those anywhere in the world. They're very easy to ship, they're very small, and then on site all we need to do is to take those eggs, put them in a box, add water and some diet, and we can produce those male mosquitoes anywhere that they're needed. So we are really excited because we think that this is going to make the technology very accessible, even in places where infrastructure would make it harder to get other mosquito control technologies into the places where they're actually needed.
That's great. Thank you, Nathan. The next question, Andrea, I'll you and where is the project in the key zone?
I think that one was for Andrea. I think Meredith's connection may be having difficulties, but Andrea, I think the question is, where is the funding for the project coming from?
Okay, yeah, funding for the project is all from the Oxitec side. There is no cost to the taxpayers of Monroe County to participate in this project. So we're very excited, like I said, to move forward with this project, seeing all of those results that have coming through from Brazil. And luckily, it's a great partnership that we have with Oxytech, and they will be footing every piece of the bill.
Okay, I think Meredith, I don't know if your connection is still having difficulties. Okay, Meredith just sent me some of the questions. So the next question is, how will everyone know if the data is collected properly and the assessment is thorough? Andrea, I don't know if you want to start with that one, and maybe I can follow on if needed.
Sure. Yeah, so as I mentioned earlier, we are going to have oversight in all aspects of data collection. We'll be doing quite a bit of quality control throughout the entire project. And there are very strict protocols as far as the data that's going to be collected and overseen by both state and federal regulatory authorities. So there'll be quite a few folks that have their eyes on the data throughout the entire project.
Okay, thank you. Another question is, do we expect any difference in the behavior of these mosquitoes in Florida compared to where they've been released previously in Brazil? I can take that one, I guess, and Andrew, maybe you want to comment as well, but I would just say we'd expect the mosquitoes to behave much the same. We have seen that our Aedes aegypti strains can mate with Aedes aegypti from multiple parts of the world without any difficulties. But what I would say is that each environment may be a little bit different in terms of its housing, the climate, and that may impact the behavior of the mosquitoes a little bit. Maybe what time of the day they're most active, or where they like to hide, or where they like to mate. And so that's one of the reasons that regulators like the EPA like to try and see data from multiple different locations as part of their evaluation of a technology. Okay, I think we have Meredith back again.
Yes, sorry, I had a weak connection, but it seems to be better now. I think you were just asking Andrea a question. Am I off? I'm sorry.
We just talked about mosquito behavior in Florida compared to Brazil, and we talked about how data is collected and assessed.
Okay, then I will ask the next question. And Nathan, I believe this will go to you. How do you determine how many Oxytec mosquitoes to release per week for these projects? Is it based on the size of the project area or baseline catch rates? Have you seen a minimum release rate needed to achieve suppression?
Okay, now that's a great question. It will depend on several factors. So obviously the area is important because that will influence the number of mosquitoes that we might expect to find in that area. It may relate to things like the rainfall, the particular climate, the time in the mosquito season as well. But what we found is that through several seasons of releases in Brazil, we can actually set a standard release rate, which works in multiple different times in the season, that can work with multiple different pest pressures, and that can be quite effective. And so we'd expect that the same would apply in Florida.
Great, thank you, Nathan. The next question, Andrea, this is really for both Fkmcd and Oxytec. So if you could answer first, and then I'll have Nathan also speak on behalf of Oxytec. But the question is, what happens if a hurricane comes in during the project?
So if we have a hurricane, we will remove all of the boxes from the field. We will remove all of the traps from the field, and really just put the project on hold until we get the all clear to move forward with from the EPA. And it's similar to what we do with our normal district operations. We won't leave anything out in the field to be blown away or swept away. And I think that that's part of the stipulation in that experimental use permit as well.
That's also one of the reasons that we have multiple sites planned for this project as well, so that if we were to have an impact on one part of the Keys, we would still have the ability to collect data if we had another part that was not affected. But obviously that would depend on the path of a storm and various other factors.
Thank you, Andrea and Nathan. The next question, Nathan, I'll send to you. And the question is, are you planning to perform similar projects in endemic countries in Central America and the Caribbean?
Okay, great question. Yes, definitely something that we're interested in. And so we are in discussions with a range of different locations. And we're really interested to get this technology into as many different places where it can have a positive impact on the wild mosquito populations.
Great. Thank you, Nathan. The next question, well, Andrea, this is really for both Fkmcd and Oxytec. So if you could start, and the question starts with your name. Andrea stated last week that tampering with these boxes was a second-degree misdemeanor. Are these boxes property of Fkmcd?
So the boxes are not property of Fkmcd. They are Oxitec boxes. I'm sure they'll be taking their boxes with them once the project is over. So no, they are not Fkmcd property. However, what I was referring to is really the state statute that basically says that you can't tamper with mosquito control operations, and because we are a part of this project, it would apply.
Thank you, Andrea. Anything to add, Nathan?
No, nothing to add there.
Okay, thank you. The next question is also for both Oxytec and Fkmcd. If you could start, Nathan, this question is, what do you think is the greatest challenge in the next steps of the Fkmcd project?
Okay. I think really the main focus for us in the next few weeks and the upcoming months is the community engagement part of this project. So really making sure that everyone who needs information has that and can ask and have any questions answered that they might have, so that when we actually get to moving these boxes and the traps out into the field, we are really ready to do that as effectively as possible. So that's a really key focus for us right now as we get going.
Thanks, Nathan. Anything to add, Andrea?
Yeah, I mean, my mind goes to, actual project deployment. And I mean, when we're doing field trials of any type, there's always a laundry list of things that you don't think of that, could affect your project. So I'm thinking, weather and, other related things that are out of our control. So I think it's a great focus that we're really looking at getting as many folks involved as possible in the project and looking throughout the keys for everyone's participation.
Great. Thank you both. Nathan, I'll send this next question to you. And the question is, we have heard of your work in Brazil, but have you only worked in Brazil or have you released in other countries?
Okay, so we have only released this mosquito in Brazil so far for the last 2.5 years, but we have released the earlier version of Oxytec's technology, the first generation mosquito in Brazil, but also in the Cayman Islands, in Panama and Malaysia, and then also done cage trials in other places as well. But we've also actually released Oxytec mosquitoes in the US already. So we've done trials with 2 different moth species, which are agricultural pests, as far back as 2006 in Arizona, and then more recently in around 2017 in New York State as well. And so we've actually released in several different countries in the world now, and we have very positive regulatory approvals in all of those countries as well.
Thanks very much, Nathan. We just have 3 questions left. We may go slightly over, but we are going to go through these 3 remaining questions, and then we'll end there. The next question, Andrea, I'll send to you. And the question is, the Fkmcd stated that they will take the COVID situation into consideration. What does that actually mean?
So we're in discussions frequently with Dr. Zuba, who is a physician on our board, who gives us updates on COVID in Monroe County. And really, the biggest question for us is, will COVID impact this project operationally? So are people uncomfortable with our employees or Oxitec employees coming onto their property on a regular basis or coming into contact with them? And that's really, I think, the main question as we move forward in the project is just, where are we in the county? Where are we with vaccinations? And how comfortable are people participating in the project with whatever's going on currently with COVID?
Thank you, Andrea. The next question, Nathan, I'll send to you. Mm-hmm. Do these so-called Oxytec mosquitoes demonstrate any sort of swarming activity after they emerge from the box? In other words, if I live near a release box, will I observe any type of mosquito-related activity that I have not seen before?
Interesting question. So Aedes aegypti is not a species that exhibits strong swarming behavior, and it tends to mate in smaller groups than other kinds of mosquitoes. But mosquitoes definitely can aggregate, usually in sort of dark or damp environments. So I think it would depend on the specific environment. But the important thing to remember here is that we release only male mosquitoes. And so they are not going to be looking for humans to actually bite and take a blood meal. That's only something that females do.
Thank you, Nathan. The next question is our last one. And Nathan, if you could start, but Andrea, if you could answer too. The question is, who actually approved the trial formally?
Okay, so the trial is approved at multiple levels. So at the federal level, this was assessed by the Environmental Protection Agency, and they carried out a very extensive evaluation of the mosquito, the technology, safety, both to humans and to the environment. And they approved this last year in April. And then following their approval, this was then considered by the Florida state authorities. So that's the Florida Department of Agriculture and Consumer Services, but bringing together a total of 8 Florida state agencies, including the Department of Health, Fish and Wildlife Conservation Commission, and so on. And so those are the federal and state approvals. And then I guess, Andrea, if you want to talk about the local district approval as well, that'd be great.
Sure, yeah. So once the federal and state approvals were in place, then it was considered by our board of elected officials, who then gave their approval this past August to move forward with the project.
Great. Thank you both. That is all of our questions, finishing almost exactly on time. Thank you all for your attention and time and interest. We invite you to visit our websites, and if you do have questions afterwards, you can send them to florida@oxytech.com and we will answer them. Thank you very much, and hope you can join us for our future webinars.